can I have one lovely volunteer tell me, what is a chemical reaction? What did we write down?
I put a chemical reaction is something that changes, and it might change in its physical form.
Okay, so we see a change in physical form. I like that.
I said a chemical reaction is a process in which two or more substances interact and something changes, but it can't be reversed.
Perfect. So two more substances interact and change, and it can't be reversed. All right. Any other ideas of what we can put down for a chemical reaction? Yeah, so I like this. So we said that we see a change in the physical form, and we also see two or more substances interacting and changing, and then that change can't be reversed. So that's super important to note. Let's look at these examples before I ask you guys for some ones that you came up with. So the examples I gave you where we see an apple core turning brown when it reacts with oxygen in the air. So maybe this has happened where you take a bite of an apple, you put it on the counter, you walk away to play video games, and then 2 hours later you come back and you're like, ugh, my apple's all brown, right? So that is a chemical reaction. That is a physical change that has happened in that apple, and it's something that can't be reversed. So we can't unbrown that apple. You can maybe scrape off the outside, but it's not going to taste the same as it did before. Same thing with an egg. I know Ashley said she's heard of this example before. When you fry an egg, it starts out as a liquid, and then when you put it on the skillet, it's going to turn into a solid, right? We're going to see a change in color. We're going to see a change in texture. And even if we try to cram that egg back into the original shell, it's not going to be the same as it was before, right? We can't unfry it. So these are chemical reactions that are permanent reactions. They're permanent changes that can't be reversed. What are some other examples of chemical changes that you guys came up with?
I put rust from iron.
So iron plus oxygen yields rust. Yeah. Anything else? Any other examples?
I said burning wood.
And Diego, I saw your hand. What did you come up with?
Baking a cake.
Baking a cake, yeah. So if we put liquid batter in the oven, so plus heat, we'll end up with a solid cake. So we see a change in substance there. All right, any other ideas, any other examples?
Also photosynthesis.
Yeah, absolutely. Photosynthesis is another example as well. Perfect. So today we're going to be talking about chemical changes and chemical reactions that we see using indicators. But before I tell you guys what an indicator is, can I have one lovely volunteer read me this introduction paragraph right here? Any volunteers? All right, can I have, Christina, will you read that passage for me, please?
Nutrients may be classified as carbohydrates, sugars, and starches, lipids, fats and oils, and proteins. Carbohydrates are a group of organic compounds that contain atoms of carbon, hydrogen, and oxygen in the general formula of H2O. The biological process of respiration breaks down carbohydrates to yield energy for plant and animal activities. Lipids provide organisms with a concentrated source of energy, yielding twice as many calories per gram as carbohydrates or proteins. Proteins are complex organic compounds formed from long chains of amino acids chemically linked together. In this exercise, sugar, starch, fat, and oil, and protein will be tested using indicators.
Perfect, thank you very much. So I told you guys that the focus of today's lab is going to be indicators. So what are indicators? Well, in food analysis, indicators are used to detect the presence of specific organic or inorganic compounds, such as sugars, proteins, starch, glucose, or oil. So today we're going to be using 4 indicators that you guys need to write down on your paper. So if you're typing, you can just type this out. If you're writing it by hand, you can do that as well. But the 4 indicators that we're going to be working with today are all right. We're going to write them in this chart right here. So the very first indicator we're going to be using today is something called Benedict solution. So it looks like this. It's blue. It's liquid. And Benedict solution, I'll write down right here, is an indicator for glucose. Okay, so here I've got my jar of glucose. We're going to be testing for the presence of glucose using Benedict solution. Another indicator is something you may have heard of before. We're going to be using iodine. We also call this Lugol solution. Right, so iodine or Lugol solution is going to be testing for the presence of starch. The third indicator we're going to be using is kind of a special indicator. It's a little bit different because we have to use 2 different compounds in order to create it. So we're going to call this Biuret solution or Biurase. And with Biuret solution, we're going to be testing for the presence of protein. And I told you that Biuret solution looks a little bit different because we actually have to use 2 different compounds. We have to use KOH, which is potassium hydroxide, and CuSO4, which is copper sulfate. So we have to use these 2 different compounds right here, copper sulfate and potassium hydroxide. And the last indicator we're going to use is something that you guys may have actually seen in your everyday life. It's something called a brown paper test, right? And do you guys know what we're going to use brown paper to test for the presence of? Do we have any guesses? So think of, oh, go ahead.
Fat.
Yeah, like fats or lipids. So think about if you go and pick up some fried chicken and you put it in a paper bag and you carry it home, what's going to happen to that paper bag by the time you get home?
It gets like greasy and darker.
Yeah, it's going to be greasy, right? So we use brown paper. Let's see a nice brown color. That's a good brown. We use brown paper to test for the presence of lipids. And today we're going to be using oil. All right, any questions about these 4 indicators before we get started? All right, take about 10 more seconds to write that down. So Benedict solution is the indicator for glucose. Iodine or Lugol solution is the indicator for starch. Biuret solution is the indicator for protein. And with Biurets, we're going to use copper sulfate and potassium hydroxide. And brown paper is the indicator for lipids or oils. Off the screen so we can get started with our lab. So on the next part of your worksheet, you have 4 places to write down the results of today's lab. I'll be periodically referring back to this worksheet to tell you guys what you need to fill in. But as I go through this lab, make sure you have your worksheet handy so you can fill in those results. Okay, so the very first indicator test we're going to do today is, wait for it. All right, very first indicator test we're going to do today is Benedict solution. Now, can someone remind me, what does Benedict solution test for? What are we going to use this to test for the presence of?
Glucose.
Glucose. So the first thing I'm going to do is I'm going to put my Benedict solution into my test tube. So we can see that the initial color, or the starting color, is this lovely blue. And when I get my glucose and I add it to this test tube, what do you think is going to happen? What do you think we'll see? Throw out some guesses.
Maybe foaming.
Maybe some foaming. Okay, I like that. What else?
Maybe it'll change color.
Maybe a change in color. Any other ideas?
Wait, I didn't hear which one that you're testing. Which one are you testing right now?
We're doing Benedict solution first. We're going to add glucose, or this is going to test the presence of glucose. Okay, so we've got two good ideas. We said maybe some foaming, we said maybe a color change. But before I show you guys the results, one quick comment. Remember that we have to have our cameras on during class. So this is now a school policy. The only way, or the only reason you shouldn't have your camera on is if your guardian has sent me an email. And so far, I don't believe anyone in this class has. So if you are not one of the people who sent me an email, go ahead and turn on your cameras. All right, so we've got two guesses. One person said we might see a color change. One said we might see some foaming. Let's see what happens. So I'm going to add my glucose to my Benedict solution. And I don't see anything happening. Do you guys have any ideas why we didn't see a change? So what if I show you what's happening right here on my table? Do you guys see anything that might give you a hint, maybe right here? All right, so this is a hot plate. Someone said they can't see, I'll tell you. This is a hot plate, and I actually have some boiling water on top of it. And the reason is because Benedict solution actually requires heat in order for that reaction to occur. So what we're gonna do is I'm actually gonna put my solution inside of this boiling water for a few minutes, and we're gonna let it do its thing. We'll come back to it in a few minutes once we see that that reaction has taken place. But while that is heating up, let's move on to our next indicator test. So the second indicator test I'm going to do is iodine, or Lugol solution. Now, can someone remind me, what does Lugol solution test for the presence of?
Starch.
Starch, perfect. So the starting color of iodine is going to be this pretty amber color. And now I'm going to get my starch and add that. But before I add my starch, I actually have to shake it up, because if you can see, the starch is all settled to the bottom of this container. Why does that make sense? What do we know about starch that would explain that? So is it a small molecule or is it a big one?
It's a big one made of simple sugar.
Molecule, right? So it's settled at the bottom because it's so dense and so chunky. So what we have to do is we actually have to shake it up so that starch gets mixed around the entire solution. All right, so now I'm going to add my starch to my iodine. What do we think is going to happen? What do you think we're going to see?
Change in color.
Change in color, let's find out. So I'm going to add my starch. And what did we see happen?
It's black.
Turned black. So that's the first thing I actually want you guys to write on your paper. I'm going to share my screen again. All right, so when, wait for it. All right, so for iodine solution, or Lugols, we're going to see it turn from an amber color, which is that orangish red. Oh no. It'll turn from an amber color. Oh, come on, I don't want brown, I want amber. From an amber color, third time's the charm, to black if starch is present. Okay, so we saw that iodine turned from an amber to a black color. Now, while we were running that test, I actually had to remove our glucose test from the heat because look, we saw a color change. So it turned from blue to orange. Now, Benedict solution is a little bit different. So not only do we have to heat it up, it also gives us a range of colors. So we actually see results in kind of a gradient. So with Benedict solution, if it's a positive test, it will turn from blue to a color ranging from red, orange, yellow, or green. So we could see one of 4 colors with Benedict solution. And if we actually see a red result, that means that there is a high concentration of glucose. And if we see more of a green shade, that means that there's a low concentration of glucose. Okay, so since we got an orange color, what does this tell us about our concentration of glucose? Is it higher or lower?
Higher.
Right, so this tells us that there was actually a high concentration of glucose in that solution because we saw it turn from blue to orange. Great job. All right, so now we've got 2 out of 4 indicators done. Before we move on, I do want you guys to put a star next to Benedict solution and write down that this is the one test that requires heat. So Benedict solution won't change colors unless you heat it up. All right, I'm going to stop sharing my screen now so you guys can see me do the next indicator test. All right, the third test we're going to do is Biarets. And remember I told you that with Biuret solution, we actually have to kind of create our own substance. So what we have to do is we first have to add copper sulfate, which is the CuSO4. That's going to be our blue substance. And we're going to add just a few drops to our test tube. Okay, and the next thing we're going to add is potassium hydroxide, which is KOH. Now everyone keep your fingers crossed because I'm almost out of this base. And if we don't have enough, we're not going to see our color change. But we're going to keep our fingers crossed. So we're going to then add our KOH, which is our base. All right, so make note that the starting color is this nice blue shade. And now Biuret solution is detecting the presence of which biomolecule? Someone shout it out. Starts with a P.
Protein.
Protein. So I've got my protein here, and be thankful that you're not in this room because it smells like rotting meat. Okay, this protein smells disgusting. And it looks pretty gross too. It's this nasty yellow shade. But now, let's see what happens when I add my protein. Okay, so nothing yet. I'm gonna add the last bit of KOH and hope for the best. There we go. So when we added our Biuret solution and our protein, what did we see happen?
It turned a darker shade of blue.
Yeah, it turned like a dark purple color. So I'm gonna switch back over to my iPad so we can write that down. So with Biuret solution, we'll see it turn from a blue color to violet, which is a light purple. So today it was a little bit darker than usual. It's kind of more of like an indigo color, but usually we'll see it like a purple or light purple violet shade. Right, so that color change tells us that protein is present. Now time for our very last indicator test. our brown paper. And can someone remind me, what is our brown paper testing for? What is that an indicator for? Oils or lipids. So I'm going to take this piece of brown paper, and starting out, it's this lovely brown shade. And we're going to call this opaque because no light can pass through it. But we're going to see what happens when we add oil to the surface. Like I said, you guys have probably seen this before whenever you get fast food. Okay, but whenever we add oil to the surface, ooh, I added too much. We see that that grease starts dripping through the paper, right? And this turns into what we call translucent, meaning that light can now pass through that paper. All right, so it looks super greasy. So that tells us that there is oil or lipids present. Okay, so let's write that on our papers. So what I want you guys to write down for your brown paper test is that, where is my brown? A stain will remain on brown paper if fat is present in the food. So the way we represent that is your paper will turn from opaque to translucent. All right. What questions do we have about indicators? Perfect. So now I want you guys to make sure you have this information written down on your paper, because the next thing you're going to do is you're actually going to complete your exit ticket. And your exit ticket today is going to count as your lab conclusion. So as soon as you have written all this information down, I want you to click on this link below, which will take you to your exit ticket. So like I said, this is your lab grade. So whatever you score on this will be your lab conclusion grade for this assignment. So go ahead, and I'm going to let you guys leave early today to work on this conclusion section. So I'm going to stick around on this Zoom to answer any questions that you might have about indicators, but use the rest of this time to go complete that exit ticket conclusion. You do have homework tonight in addition to completing this conclusion. You need to watch your Enzymes Day 1 video and submit your notes before tomorrow morning. Right, what questions do we have? Perfect. If you do not have any questions for me, go ahead and log out and get started on that exit ticket. You have to submit your conclusions before 3 p.m. today. So get those done right now. You've got plenty of time to finish it. All right, have a wonderful day, guys, and I will see you tomorrow. Again, I'm going to stick around to answer any questions you have about this assignment. Bye. Michelle, I see you said, can I see the note again? Which note are you talking about?